What is statistical parametric mapping? 4. Brain map interpretation. Imagine being able to see exactly which parts of the brain light up during different activities or conditions. That's where statistical parametric mapping comes into play. It's a method used to analyze brain imaging data to find areas of activity or change. When scientists or doctors look at brain scans like functional magnetic resonance imaging or positron emission tomography scans, they need a way to understand what's happening in each tiny part of the brain. That's what statistical parametric mapping helps with. This technique works by assigning a number, called a statistical value, to each small volume of the brain, known as a voxel. These values tell us how likely it is that a specific brain region is active during a task or condition. The process uses a model called the general linear model to connect brain signals with what the person is doing or experiencing. This helps measure how brain activity varies with different stimuli or tasks. Once the statistical values are calculated for each voxel, they are combined into a map called a statistical parametric map. This map visually shows which areas of the brain are more active or different between groups. But because brain scans contain thousands of voxels, there's a risk of false positives, finding activity that isn't really there. To prevent this, the method uses a statistical approach called random field theory. It adjusts the thresholds so that only truly significant activity is shown, making the results more reliable. Most of the time, researchers use software built in MATLAB to perform these analyses. The software helps with preparing the images, aligning them to a standard brain, smoothing out noise, and then running the statistical tests. The final maps produced are then interpreted to understand brain function or diagnose problems. In practical terms, statistical parametric mapping allows scientists and clinicians to see exactly which brain regions activate during specific tasks. It helps compare different groups, such as healthy people versus patients with neurological conditions. It can also reveal changes in brain activity caused by diseases or treatments. This makes it a powerful tool in brain mapping and diagnostics, providing clear, localized information about brain activity. It's a key part of how neurotechnology advances our understanding of the brain and helps improve diagnosis and treatment options.